Finally, in this morning's session is Dr. Peter Bandettini. He's currently director of the NIH Functional MRI Core Facility and chief of the section on functional imaging methods in the Laboratory of Brain and Cognition, of which I'm part of that alma mater, and at NIMH. And he was past president of the Organization for Human Brain Mapping. He's received the Scientific Director's Merit Award and the Wiley Young Investigators Award. He received his PhD in biophysics from Medical College of Wisconsin, where he and Eric Wong played a role in the early development of blood oxygen label-dependent functional MRI, which is now really a widely used technique. And he continued that developmental work at Mass General Hospital before moving to NIH. At NIH, his laboratory continues to develop fMRI, and the work, he considers fMRI has untapped potential for revealing a wealth of neural and physiologic information from individuals. And as we'll see today, he's now becoming interested in using this wealth of information to guide stimulation and neurofeedback. So Peter's talk is entitled Functional MRI of the Individual, from Biomarkers to Neuromodulation. So join me in welcoming Dr. Bandettini.
Okay, thanks Vince. And thanks for inviting me to this. I really do appreciate it. Actually, I was planning on attending, and I was asked then by Vince to speak a little bit. And my thought initially was I'm an fMRI methods guy, and that's my expertise. And so I'm sort of going a little bit out of my comfort zone in terms of talking about this. But I think actually this is where I feel pretty passionately about where fMRI is going. We're going, very much away from group studies. Group studies, having large databases is important, but I think it's ultimately important for building classifiers for individuals, for putting it actually in the clinic. And so that's actually what I'm going to be talking about. How do we actually go from group studies to individual studies with fMRI? Okay, so my talk will be two parts. One is using fMRI or MRI actually for individual biomarker development, and just sort of general concepts, sort of stepping back and sort of give a general concept on where I think the field should be going in that regard, and where it is going actually. And I'm cheering as much as possible and trying to jump in where I can. And then secondly, neurofeedback and neuromodulation. And even though they're for both number one, still, that's just... Okay, so to begin, this is a chart that my former co-advisor, Scott Hinks, who's actually a physicist at GE, gave me when he was giving a talk about how GE thinks about things. But I think it's really telling. I think that... so basically, these are various disorders, and there's several even left out here, like schizophrenia and things like that, versus MRI adaptation or adoption on how they're assessed. And so typically when you think of an MRI, you think of, looking for tumors, doing routine brain for any sort of, hitting your head or something, maybe a little bit for MS, things like that. So epilepsy, it's sort of moving in that direction. But there's all kinds of things that have a huge potential for assessing depression, Alzheimer's disease, stroke, TBI, huge potential. They're still barely adopted. And that's a real sort of bottleneck in terms of and a huge potential. So and there's also this trend, and I was actually just talking to Peter about this morning. Is Tom Insel is sort of putting forth this concept of it's called the RDoC, and it's basically the idea is going away from DSM, sort of having physicians categorize people with various disorders, having rather the disorders sort of categorize themselves in terms of actual measurables. And the common analogy is sort of if you have a heart problem, you actually look at the heart to try to figure out what's wrong, as opposed to try to look at other evidence and sort of use your intuition. I mean, certainly that's helpful. That's information, but there's a lot of other information that can be had. And so the idea here is that if you diagnose someone with depression, they could be any one of several different categories of genetic risk and brain activity and physiology. But if you actually divide them into these categories based on their specific measures, then you can make a more precise diagnosis or maybe a more precise prescription So that's the goal of, and imaging fits in there in terms of either helping to diagnose or actually helping to predict what will work or not, sort of having a more precise brain activation pattern. Okay, so let's take a few steps back. And also importantly, actually, when I was talking to Tom, he actually mentioned that, this goes all the way up to Congress. Congress looks at fMRI and says, we've been doing group studies for 25 years now. What have we gotten? Certainly we understand the brain better, a little bit better, but what can fMRI do to actually help patients? And so we really need to start putting people in scanners in the clinic to derive information in individuals. So how much can we get from using MRI and fMRI? So just to start, we can certainly see activation in individual single subject runs. We can see the motor cortex very easily and all kinds of other things. We can see modulations of that activation. We can see differences in activation that are correlated with performance and behavioral and perception. So we can actually see how that correlates with their performance as well, which is pretty good. And we certainly can also go beyond that. We can now, using multivariate methods, we can decode what they're viewing. So for instance, if they're viewing a movie or whatever, we can easily see that, or somewhat easily see that within an individual subject. So old movie that I like to show, this is decoding by eye. We look at this as an axial movie. This is back 1991 myself. And it's pretty clear to most of us that what I'm doing there is moving my left fingers, then my right fingers, and so on. And so obviously decoding methods become much more sophisticated than that, since the activation isn't as obvious of a motor cortex activation for most other things. So what can't we see in that movie, though? So you might be able to, so in terms of drawing inferences about that subject, though. Sure, you can tell what they're doing, but what we want to do is draw inferences about the subject. So there have been fMRI studies on looking at handedness or language dominance that might replace the WADA test. That's certainly something. That's one step. But there's been other directions as well. So we'd like to, though, derive information about biologic age, gender-related differences, sensory motor characteristics, differences in cognitive and personality traits, psychological state, physiologic state, neurologic differences, of course, and also developmental differences. We can, I believe, pull out these differences from a combination of MRI and fMRI. It's just a matter of finding out the features to look for and getting clever about our classification methods. So this is the direction I think we need to be going in terms of pushing individual subject assessment. So why has MRI been extremely successful clinically? So pretty much fMRI has been riding on the back of the fact that industry has a lucrative market in MR scanners, but it's not for fMRI, it's for things like just looking at tumors or things like that. It's been incredibly effective. And here's why. Pretty much most of us who are non-radiologists can just look at this and see it. And but more accurately, there's an effect size to noise that is incredibly high. It's a factor of 10. And of course, there's where the radiologist's eye comes in is where things are much more subtle and where they learn to look for much more subtle things. But even then, you need to have a pretty robust effect size to noise. We also have a clear gold standard. We know where the tumor is. So with fMRI, for instance, if we try to diagnose ADHD, for instance, with fMRI, we don't really have a clear gold standard of what the ADHD is. And so back to the RDoc sort of initiative, we should try to have either genetic markers or things like that that might help guide the imaging, the fMRI classification. Okay, and so here's the effect. If you have an effect size to noise of 10, you have two distributions of group effects, they're huge. And so any individual, if they would fall in one group or the other, is clearly able to be binned in that group or the other. It's very easy. But if they're like this is most group studies with fMRI. Certainly you have, if you look at the average, clear effect, that's very robustly different. But if you look at the distribution, if you take any one subject, one component of this distribution, it's hard, even for the ones that are on the far end of this distribution, to say with enough certainty that that subject lies in this distribution or it should be classified within this other group. So that's the problem that we have here. So But just before I dig into that a little bit, I just want to sort of re-emphasize the point that there's been a ton of studies, and I'm just going to show a couple that really show that MRI-derived information or fMRI-derived information are exquisitely sensitive to individual traits. Here's one example. This is some work by Jeff Boynton, his group, showing basically mapping out using fMRI the visual cortex, V1, V2, and so on, and actually finding that there's, without going into too much detail on these measures, but these are various measures of visual acuity, and measuring and showing that there's this relationship. It's not very clean, but it's there of between size of visual cortex and visual acuity measures. Another example, predicting individuals' learning success from patterns of pre-learning MRI activity. Looking at the dorsal striatum, people have found that, groups have found that you can predict how well they'll learn a video game. This is looking at the activation looking at a video game. And there's other ones too. You'll predict how well they learn this by specific activation in the dorsal striatum. This is not MRI, but it's sort of just another example. This is using EEG, also showing prediction of learning a motor skill based on the coherence around motor cortex. You can use spectroscopy and look at very subtle differences. This is measuring GABA, and looking at GABA in very specific areas of the brain correlate quite nicely with measures of impulsivity. So, or urgency, or sense of urgency in the subject. So MRI, structural MRI can look at subtle differences. This is a really nice review article. I'm just showing some examples. Differences in visual choice reaction time, response conflict, differences in speed accuracy, trade-off ability, all seem like they have anatomic correlates that seem to show nice trends. So these are subtle differences that show up in very subtle, very specific anatomic regions. Also in conscious perception, visual illusions, ability to see illusions, even metacognition show relationships. So they're all there. So there's MRI and fMRI are sensitive to subtle effects. It's a matter of pulling in these distributions such that we can then say something about individuals as opposed to just looking at trends. So not going to go into this. This is a field that I think is exploding in fMRI. It's already exploded elsewhere, but the idea is to classify data. And so you need a large training data set. It's hence a big use of a lot of our databases that we are acquiring. I think that we need to actually have those databases more well curated. So, I mean, there might be very subtle differences, but you should really try to have the data collected in almost exactly the same way. There's enough vendor-specific differences, there's enough pulse sequence differences that would really add noise to the data. So that's a goal, to sort of have more curated data sets, even behaviorally better curated data sets as well, the behavioral measures. So then from those training data sets, you try to figure out which features to extract. And so this is sort of an art in itself. I mean, is it just fMRI area? Is it latency of the response? Is it magnitude? There's about 100 features that you could extract that may or may not give you information. It's nice to have that sort of data-driven as well. Minimize or better characterize the noise and variability. So that's where having gold standards comes in. And also trying to reduce the noise. Like we have a sequence that we've been pushing multi-echo acquisition that reduces the noise in a time series such that you can actually pull this information out more easily. Design paradigms to maximize the effect size, and then finally train your model and optimize it such that then you can apply it to test data or even apply it clinically. So this is sort of the pipeline that I think that we can potentially apply to a lot of our studies that we're doing right now. And so just to show you, I mean, as an example, what measures can we obtain with fMRI and MRI? We can obtain all kinds of measures. So there's, location, extent, magnitude, shape, latency, undershoots, transients. They all might contain information that could be used in classifiers. Dynamics, connectivity, dynamics of connectivity, and so on. In the anatomy, everything from gray matter to gyrification, to myeloarchitecture, all this is potentially fodder for classification. And of course, spectroscopy has many metabolites. So, and also the sources of variability, not only are they are MRI related, and the goal is to actually make this curve as narrow as possible, but they're related to the subjects. So here's just one quick example that I like to always bring up is that, when we look at group studies, we see this average result, but we actually, across individuals, there's this distribution. This is some nice work by Mike Miller. And you have this distribution. And is it noise? Well, just one example of one subject that showed a very different distribution. This is sort of an episodic memory retrieval task. This is the group analysis. This is the single subject. And you might think, oh, this is just noise. But in fact, this subject did have a very stable, different distribution of activation, even 6 months later. So what does this mean? So this subject, either this is a variable that we need to control for, or there's interesting information in this subject. And so trying to understand why they're different in that regard is also a direction that things could go as far as that's concerned. So how do we extract these robustly? Well, of course, with machine learning, we can't just use one dimension. It's a multivariate sort of approach. And so there's a nice review article by David Linden on exactly this type of thing. And the basic concept is that I'm sure everyone in the room is familiar with is multivariate classification, multidimensional classification, where if you take just one distribution, it's impossible to separate them. But if you take a different distribution, this happens to be sort of a model system of looking at hippocampus volume, looking at Alzheimer's disease. You can't differentiate just looking at one distribution, but looking at two, you can. You can draw a plane that completely separates those with Alzheimer's versus those without. It's not this simple. You might have to have 10 variables that you might be able to draw a hyperplane through them in that way. So that's the basic concept of trying to have several variables that you're looking at the same time and trying to then classify and pull apart such that you have no overlap into distributions. So there's already been groups that claim they can do this pretty accurately. This is a recent paper saying that brain images alone can accurately diagnose chronic neuropsychiatric illnesses. This is ADHD versus this is Tourette's syndrome and healthy controls. So they found in looking at various structures of the brain, they found the red is sort of where they're different, they're larger. The blue is where they're slightly smaller. So using these, they're actually in a bottom-up sort of way finding features that they could use in a template to classify. Looking also at adults. This is looking at different susceptibility for depression. This is actually risk for depression. So there's looking at adult brains. And it's probably not this simple, but this is a really intriguing paper as far as looking at sort of a bottom-up approach, saying, okay, these are the areas. And not only do they stop at saying, oh, these are the areas, they actually say, oh, this is the effectiveness of these areas. So it seems like there's some really strong potential for carrying this forward. So the ultimate goal then would be to do this sort of thing. So you have the studies done here, where you have lots of different imaging centers, lots of databases. You curate the data, you come up with a classifier that classifies them, and then you can actually then distribute this to centers around the world. Obviously with the help of radiologists, they can then look at this sort of classifier to then help either make the diagnosis or plan their treatment. So that's the ultimate goal. And okay, now I'm going to completely switch gears and talk a little bit about neurofeedback and neuromodulation. So as has already been said this morning, very, very well, there's many uses for fMRI and MRI in the context of neurofeedback and neuromodulation. And that's either detailed multimodal registration. So you want to register your fMRI and anatomic data with each other, but also such that you can actually have the probe sort of properly aim. Real-time feedback for therapy. This is using fMRI on an individual for real-time sort of feedback, either fMRI feedback or neurostimulation feedback. And, of course, neuromodulation guidance and assessment. So what's unique about fMRI and MRI is that you obviously can guide where you put the stimulus, but you can also get a sense, based on the activation, not only looking at behavior of how effective the stimulus is. And also you can sort of start to probe connectivity as well, as was mentioned. So just to start, detailed multimodal registration. this is some really beautiful work that Ziad Saad has been developing in the context of apnea. And that is, so you can't really get this sort of data looking at average subjects. It's only with individuals. And the problem with this type of data is one of visualization. And so he's struggling with apnea to try to figure out the best way to visualize it. And really the answer is you'd have to have it easily manipulatable by the user. So here's just an example of track tracing combined with anatomy, trying to sort of look at it the best way possible, because the devil's actually in the details in terms of looking at exactly how these connect. You can also combine simultaneous fMRI. In this case, it's resting state, looking at these various nodes and looking at their correlation over time. And also at the same time, you can look at how they relate to connectivity with fiber tracks and also the anatomy. So having all this data displayed simultaneously, I think is still useful for the clinician or potentially the user who really needs to probe this data, because it's so difficult to sort of collapse ahead of time into a digestible sort of piece of information that you still need a user interface to sort of work with the data. So that sort of has been something that Ziad has been working on. This is another example. And here's one more example of actually just looking at a seed voxel correlation with activation. So here's the activation, and then you can move this ball around to look at not only the fiber tract connectivity here, but also how this area correlates with other areas in terms of, and I have a quick movie just summarizing this sort of thing. So this is, like I said, this is Ziad's work. So you can actually in real time move around a seed, look at DTI tracks, but this is already too much to take in. So how do you do that? So you start to figure out ways of manipulating the brain, and so you can make it transparent. Here's a seed looking at the connectivity. And from there, you can actually then split apart the hemispheres to sort of look at it better. And I found this fascinating because I think that there is, so actually this is looking at the fiber tract connectivity to those seeds, every fiber tract that goes through this seed, and also the functional connectivity with that seed. So it's a nice way, immediately you start getting potential insights as to how things are connected that you wouldn't just see by having this data all at once just shown. So, okay. I'll just go on. Oops. Oh, oops. Oh, there we go. Okay. And on top of that, I actually think that there's room for development for what's I like to call neurosignatures. So certainly fMRI and PET are very difficult to have a subject walking around mobile doing whatever while you're doing them. So what we'd like to do is actually do EEG simultaneous with fMRI to develop, not solve the inverse problem. It seems like already we've been agreed upon that's pretty hard to do, actually, even with fMRI, because it's a very fragile sort of model with using localized areas with fMRI to help solve the inverse problem. But what you could do is find an fMRI pattern that corresponds to an EEG response, an EEG signal. So you have an fMRI signature of that EEG response that you might be able to then put into a classifier. So what you could do then is have people walking around wearing their EEGs, doing sort of free behavior, and you know really what the underlying activation is based on how well these map these templates that correspond to the underlying activation. And yes, you might have non-unique solutions, but they might be unique enough if they're well characterized enough. Same thing with PET and fMRI. You might have PET scanner just looking at dopamine release, and you can't do that with fMRI, but you can find the fMRI signature of dopamine release, or you might even be able to find the EEG signature of that as well. There might be some unique signature. So the whole idea is to try to find, using more cumbersome techniques, the information, but then finding the signatures with more collapsed data, skeletal data. So real-time feedback for therapy. I think fMRI, one other clinical use for fMRI with individual subjects, I think is growing, is real-time. So for a while, there were some false starts. Real-time was a cool thing. You could do it. Feeding the data back to the subject was nice. But at the same time, I think that There may have been some false starts, but right now it's coming back. I think that especially Rainer Goebel, he's been doing some really nice work in developing the paradigms for real-time feedback. So the idea here, you have the subject in the scanner, the images are reconstructed, they're collapsed in some way that is informative to the subject, that feedback is fed back, and they're asked to try to modify their brain activity. And what's, I think, key here is that so many people have been trying to do real-time feedback in a continuous fashion. So the subject is like multitasking, which seems like a mess, where they're constantly looking at the feedback and trying to sort of modify their behavior, which is not the same sort of task. So Rainer sort of settled upon this type of task, where you actually have 20 seconds of just doing nothing, and then you have 20 seconds of just trying to regulate what you're doing, and then you wait and you look at the lagged response to see if you did it right or not. And so having it more in blocks, it seems to make it more effective. So a couple of examples of how this has been very effective. One is for treating patients with depression. So the goal here is to upregulate previously found brain networks corresponding to emotional activation. And basically, this is across 4 sessions. They were pretty dramatically able to upregulate this. And so not only did they change their brain activation, they upregulated these emotional networks, but their symptoms of depression actually went down pretty significantly across the board. This is for individual subjects. This is on average. And this is now in clinical trials in England, actually using as a therapy, a real-time sort of feedback therapy. And also, it's been performed in Parkinson's patients, having a little bit more motor control. So also feedback has been performed as well. So not only neurologic disorders, but psychiatric disorders are potentially treatable to some degree with real-time neurofeedback. And also, you could just use this. This is some nice work by de Pettencourt of just seeing if a subject is paying attention to whatever they're paying attention to. So this is looking at either faces or houses, and using a multivariate classifier of whether they're paying attention to faces or houses. They were able to pretty accurately tell when the subject was looking at faces or houses. So another nice piece of work as far as that's concerned. Okay, lastly, I'd just like to end with talking about neuromodulation and guidance and assessment. So there's been many techniques talked about this morning, but there's a whole bunch of, I sort of dug into the literature a little bit and talked to a number of people. There's a lot of techniques on the horizon that are potentially useful as well. So this covers a few. Everything from deep brain stimulation, tDCS, tACDS, rTMS, vagus nerve stimulation. But there's a couple here that are intriguing, at least to me, because they're new and very non-invasive. There's focused ultrasound, near-infrared light therapy, and what I found even most intriguing is low-field magnetic stimulation. So that, it turns out, is alternating your magnetic field in a very slower fashion, which is almost exactly what we do when we do echoplanar imaging with MRI. So maybe whenever we get MRI, we're getting some sort of neural therapy of some sort. So Holly Lisenby has been one person sort of pioneering a lot of these techniques, and she has some really nice review articles on this type of thing for treatment of depression, especially, or other sort of somatic disorders or mood disorders. So just to quickly give a quick overview, nothing really too insightful here except for, this is sort of an illustration of deep brain stimulation. And in this particular study, the focus was the subthalamic nucleus. And this is for treatment of Parkinson's disease. But as is already mentioned, Helen Magbird has been pioneering sort of the treatment of this for depression as well. And it's sort of still an art. And I just want to reemphasize this point, is that there's various ways of localizing. For instance, this is the subthalamic nucleus localized by just looking at susceptibility contrast, as opposed to DTI or MRI. This is a unique contrast with MRI that's useful for that because there's a slightly higher iron content. But it really is an art. She has some results, but it might depend very much on exactly how you place these electrodes. there might be other techniques on the horizon that are less invasive as well. So it kind of works, but it's still the story is not completely concluded as far as that's concerned. And as far as applications of deep brain stimulation, it's everything from Parkinson's disease to writer's cramps to Tourette's syndrome, chronic pain, OCD, and things like that. But I'd like to also illustrate a point as well, is that this is work by Adam Messenger and collaborators in Leslie Ungerleider's group. There will be a poster presented tomorrow in which they're using deep brain stimulation or a form of deep brain stimulation for looking at connectivity across structures. Basically, this is looking at a functional imaging looking at a monkey at 4.7 Tesla. 2 rhesus monkeys were used. Essentially, electrodes were placed in the amygdala and then repositioned. And actually, the amygdala was activated, and they were looking at the corresponding bold activation. And what they found was, depending on where they put these electrodes, they had different activation on the ipsilateral side, just focusing on the ipsilateral side, and finding that there's shape and face areas activated by selectively stimulating the amygdala. And it seems to indicate that that amygdala feedback to categories like various, the ventral visual stream is from the dorsal basal nucleus, but also that it's able to map out this sort of feedback mechanism as well. That's was new to me in the first place, and actually just emphasizing that there was a stimulus, there was the activation at the electrode tip, and basically dividing the brain in between face and objects. There was a little bit more activation to face than to object patches. So showing a selectivity to faces that feed out from the amygdala. So anyway, from that, this is tDCS. No need to go over this. This has already been talked about. I think I just want to emphasize one point here, though, and that I tried to emphasize a little bit in my question. I think that there's been great models of tDCS, but I think that there's a huge potential. Part of what I've done in the past is trying to look at neural currents, intrinsically human neural currents, and for the most part failed. But I think the field, we still need about an order of magnitude more sensitivity to see those. But I think we have the sensitivity easily to see current distributions with tDCS. And so if you map out the phase of the NMR signal as it's applied with tDCS, I think that you will be able to see pretty accurately your current distributions. It's a matter of doing it. And so I'm sort of trying to gear up to do that myself, but I think it could be potentially a very nice real-time. You can imagine in the future sort of having a real-time, sort of moving the tDCS probes and actually seeing in real time the current distribution over the cortex, which I think is very possible. And TMS, already been talked about. But one thing actually two last things that I want to talk about before I end are what really intrigued me, this idea of focused ultrasound. So there's been only a couple of papers out. Taylor actually has been sort of pioneering this. And the idea is that you have a transducer that actually can be used in a scanner simultaneously with imaging, in which you can, instead of causing activation, it makes this particular study, they're making the area more sensitized to detecting discriminating either patterns or whatever. So it's like it was making S1 more likely to fire in some sense. So, and it worked pretty well, and it was very non-invasive. And so here's the example of that. So actually having a very focused probe at S1 with ultrasound and actually looking at the either 2-point discrimination or frequency discrimination, very clearly differences are shown here, either with frequency or 2-point discrimination. So it sort of upregulates, kind of similar to what tDCS would do. Makes that area probably more likely to fire. And one other really interesting application that I just had to throw in here because it just was a little bit mind-blowing of focused ultrasound was in treatment, in therapy for Alzheimer's disease. So the idea here is that, and this is only on animal models, is that you inject their vessels with these microbubbles, which actually resonate a little bit more with the ultrasound. resonates so much, and so you have an ultrasound probe going through the brain, and as the blood passes through, you can actually focus it in a specific area. As the blood passes through that ultrasound field, these microbubbles vibrate all over the place. They break the blood-brain barrier, and that essentially allows antibodies to be released, or plaques, so the beta amyloid plaques, to be pretty much cleaned up from that. And so it's been very, in rat models, this is before the focused ultrasound treatment, this is after. The plaques are still kind of there, but they've gone down pretty much. And so people are kind of excited about this in the context of a rat model. And also, the animal showed no behavioral deficits. So the blood-brain barrier breaks, and then it seals back up again, and then the rat's fine. So it might be an interesting way of probing as a therapy. So the last technique that I found intriguing was near-infrared light therapy. And the idea there is that, and this has been around for years, essentially you have an optical window of between 600 and about 1,000 nanometers in which you can selectively shine infrared light to the extent that it penetrates the cortex. It upregulates metabolism, essentially, mitochondrial upregulation. And this has all kinds of good effects, either acute effects or more long-term effects on aiding cell proliferation, cell motility, growth factor production, things like that. So initially that seems a little bit out there. I mean, it's been around for years for, maybe probing or trying to heal wounds. People have been using it to grow hair, all kinds of things. I never thought it'd be used on actual therapy. So this is an example of somebody getting therapy, near-infrared light therapy, for neurorehabilitation and finding pretty significant post-treatment cognitive performance increases. So in the context of traumatic brain injury, though, this is showing clearly increases in, at least looking at the frontal lobes, increases in inhibition and inhibition accuracy. Another example of near-infrared light therapy showing improvements in 10 patients with depression and anxiety. So not only in cognitive performance, but also treating depression and anxiety. So an interesting sort of probe, interesting sort of thing is that the mechanism isn't exactly known, but it seems like it's somewhat effective. I'm still amazed that it penetrates the skull at all. So that seems like a kind of interesting direction. And so basically, this is pretty much going over the mechanisms, but I'm not going to touch on that just in the sake of time. More papers on using it for cognitive enhancement. So it's been out there since about 2004, but the number of papers are really taking off. And there's acute effects as well. This is some nice work on, but it's a different mechanism. It's not cytochrome oxidase upregulation. It's just thermal effects of light. So this is interesting in a sense that instead of actually using optogenetics to selectively activate neurons, they go in with infrared probes and they just give those neurons a burst of light and they become activated. Slightly differently than you would with electrical stimulation, but they still become activated. So that's an interesting effect. And the hypothesis there is that it's a heat gradient that sort of depolarizes the neurons. Another example, actually I was just talking to Anna Rowe, but another example of selective activation based on near-infrared light, looking at this heat gradient effect and actually looking at increasing spiking. So it's a very effective probe or technique. And as I mentioned, low-field magnetic stimulation, our echoplanar imaging gradients are pretty much almost exactly that. And so that might be something interesting for probing as well as far as neurostimulation. So lastly, in a conclusion for this part, for most of these methods, I wouldn't say all of them, most of them we have a pretty good understanding, but we have an obvious mechanism of action, location, or consistent outcome. So it's still a little bit out there. There's some converging evidence that these are effective, but it's not completely converged yet. So there's a lot of opportunity for research. So MRI and fMRI and spectroscopy will play a large role in understanding these mechanisms and mapping these effects and also helping to guide them as far as aiding their clinical use. and also producing more consistent results. This is what the last couple of talks have been already about, is that the results, they get results, but it's a matter of making them more and more consistent is part of the barrier to actually applying them clinically. So with that, I'd just like to end. All right, thank you.